This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Today we're talking about antibodies and vaccines. From the first vaccine used to prevent smallpox. Smallpox is pretty horrifying. If you look at, just do a Google image search for smallpox if you want to be scarred for life. It's awful. To new efforts to understand how the immune system recognizes influenza in an effort to make better flu vaccines.
Most of our antibodies are directed at the top of the lollipop. And the problem with this approach is that this area of the virus is quite variable.
And we'll discuss how we can use our understanding of the way that antibodies work in order to make better therapies for HIV.
Every patient that gets HIV basically has a mini evolutionary cycle that goes on with the HIV virus. So there's a constant mutation battle between the immune system and the HIV virus over time as you're infected with it.
Hi, welcome to Audiommunity, Episode 2. I'm Kevin Bonham, and I'm a graduate student in the immunology program at Harvard. Joining me, also in the immunology program, is Matt Woodruff.
Hi everybody, I'm Matt.
Also joining me from the virology program is Kate Franz.
Hi, I'm Kate.
Today we're talking about vaccinations and antibodies and how we can use our understanding of how antibodies function in an immune response in order to make better vaccines or to protect people from infections in ways that are similar to vaccines but aren't exactly like vaccines. So just to give you guys some history, probably most people listening have had vaccines before, and if you haven't, shame on you. But the first vaccination is credited to a guy named Edward Jenner, who was living in the 18th century.
Whose direct descendant is Bruce Jenner.
Right.
No! I hope it's true.
And they're direct descendants of Henry Cavendish as well. So the first vaccination was credited to a guy named Edward Jenner in the end of the 18th century. And the way the story usually goes is that he recognized that people who were infected with a disease that's called cowpox, usually people working on ranches, milkmaids, would get a disease called cowpox from their association with the livestock. And those people were protected from the much more dangerous and deadly smallpox that was killing lots of people and was a constant health threat. And the story goes that Jenner realized that this was happening and thought, hey, if I infect people with cowpox intentionally, maybe they'll be protected from smallpox. And cowpox wasn't a particularly scary disease, so this would be a really great way of preventing people from getting smallpox. Smallpox is pretty horrifying. If you look at, just do a Google image search for smallpox if you want to be scarred for life. It's awful. But the other thing is it does kill you in many instances. I think it's like 30% of people infected with smallpox die from it. But even if you don't die from it, there's a pretty good chance you're going to have massive scarring and just gross deformation of your face. And it's terrifying. So smallpox is bad. Preventing it is good. And a lot of people give Jenner a lot of credit for that. Turns out he probably wasn't the first person to figure this out, though he was the first person to sort of make a big deal about it, publicize it, do sort of systematic and controlled studies to show that actually it was the infection with cowpox that was preventing smallpox. So he generally gets all the credit.
Can we talk about the likely, or the story that gets passed down about him, where he threads the needle through his son's arm because he was so confident?
It's his gardener's son's arm. It's not his own.
So yeah, if we could just publicly set the record straight. So Jenner's story is normally told where he threads the needle through the cow pustule and then he threads it through his son's arm to, nobly prove that his vaccine works. And it's very, very unlikely that that's true. It was probably one of his gardener's sons. Anyway.
No, it definitely was. The name of the boy is known to history. I forget what his name is, but yeah.
I don't know it. It's not known to me.
Actually, it's also interesting, this idea that previous exposure to a virus could protect you from subsequent infection also wasn't really a new idea. So people had known for a while that if you infected someone with actual smallpox through a process called variolation, which generally gave you a more mild disease than accidental infection, though sometimes it could turn into full-blown smallpox, this prior infection would then protect you from future infections with smallpox. And people were performing this, though there was some controversy around it for a long time. But in any case, Jenner is credited with vaccination, and actually the word vaccination comes from the Latin vacca for cow, because they used cowpox. But we now know that vaccination, which generally is infection with dead or weakened versions of a pathogen in order to confer protective immunity against the actual infection. Vaccination is super effective, and it's been developed for a number of different infectious diseases. And everybody, when they're kids, or most people, if they don't have crazy parents, get vaccinated against a whole host of infections, and they're generally pretty effective. Not 100%, obviously, but they do a really good job. And if you get enough people in a population vaccinated against disease, you actually can protect people even if they're unvaccinated or if the vaccines on them weren't effective, because you get something called herd immunity, which basically prevents an infection from getting into a population of vaccinated people.
Can we talk for a second about the group in North Texas right now that's having outbreaks of measles? Has anyone heard about this?
I heard that there was a church that...
Yeah, so there's a mega church in North Texas where one of the stances has been, you don't vaccinate your kids. And this is not a statement on religion at all, so I hope people don't take it that way.
No, actually, the biggest pockets of anti-vaccination groups are actually like wealthy liberals on the coasts.
Yeah, exactly. So it has nothing to do with that. But this just happened to be an evangelical group that has basically told their huge population not to vaccinate against normal things, measles, mumps, rubella, the MMR vaccine that everyone's given when they're very small. And so they're having a huge outbreak of measles in that town, because it turns out when you don't vaccinate all of your kids against measles, and one kid gets the measles, then everyone gets the measles.
Yeah, measles is another one of those pretty horrifying diseases. And I think a lot of people, because we've had such effective vaccine campaigns for so long, I think people are sort of divorced from really how awful some of these infectious diseases are. Things like pertussis, things like measles, they're really, really terrible. And the idea that, I don't want to go down this rabbit hole too deep, but...
Yeah, no, but just ask your parents how the mumps felt.
Or about how terrified they were of polio.
Right, exactly, of long-term paralysis. So anyway, we don't, like you said, we don't have to go down that route, but it's pretty easy to make those arguments.
Yeah. So anyway, back to the history. So Jenner figured out the vaccine, or he's credited with the vaccine, in the late 18th century. And people sort of ran with it. This idea that you could vaccinate using a killed or weakened version of a pathogen became really popular. And during the 19th century, people got much better at identifying especially bacterial illnesses. And there were a number of scientists in a number of different places in Europe that were looking at vaccinating against certain microbial infections. And towards the end of the 19th century, a guy named Paul Erlich, who was a German scientist, was doing work with several different bacterial infections. And he coined the term antibodies. Because he was German, it was Antikörper. I'm not pronouncing that correctly, but it had been known for some time that the serum, which is basically blood without the cells in it, so the liquid that your blood is flowing in is called serum, and it was known that the serum of immunized animals could kill infectious bacteria. So in other words, if you have an infection with, say, anthrax bacteria, Bacillus anthracis, and you have immunized a guinea pig or a mouse against Bacillus anthracis, and then you take a few weeks later serum from that animal, that serum is capable of killing Bacillus anthracis, but often it's not capable of killing other related bacteria. And the serum of unimmunized animals is also not capable of killing that bacteria. So they discovered that there's something in the blood, in the serum of immunized animals, that was capable of killing bacteria and was also responsible for protecting those animals against future infections with the same microbe. They were able to separate the ability to recognize the bacteria from the actual killing action. So there were some really cool experiments done in the early 20th century where they basically found that if you heated serum up to about 55 degrees centigrade, you could remove the ability of that serum to kill bacteria. But if you mixed that heat inactivated serum with serum from an unimmunized animal, you would regain the ability to kill bacteria. And so they basically figured out that there were two components of serum that had this anti-microbe activity, one that was responsible for recognizing the bug and one that was responsible for actually killing the bug. And that thing that kills it is called complement. We may get to that in another episode. But the ability to recognize the bacteria, that was caused by what Ehrlich called antibodies and what we still call antibodies today. And we now know that these antibodies are proteins that are produced by cells called B cells. And B cells have this really... I'm about to talk about VDJ, which is going to be rough.
So I'd like to point out that we do have a brief explanation up on the web. You can see what an antibody looks like, how the basics of it work, how it's formed, those sorts of things. So if anybody is completely unfamiliar with the term antibody, some of the B cell stuff that Kevin's about to throw around, please feel free to browse or peruse through Visimmunity, because we've got an assortment of things for you to look at. But yeah, best of luck with VDJ recombination, Kevin.
Yeah. Okay. So it's about to get complicated, but it's super cool. This is actually, I think, many immunologists, their first, the thing that first got them to fall in love with immunology was the idea of how antibodies are generated. Which is a process
it's also the thing, surprisingly, that causes many immunologists to fall out of love with immunology.
Also true. When you have to understand the mechanism of it, it's very complicated.
Right, exactly.
But the way that you can get so let me back up a little bit. B cells, in principle, can make antibodies against any chemical that has ever existed or will ever exist in the universe. And this seems like a pretty crazy statement, but people have actually shown that you can generate antibodies against chemicals that have never existed in nature. They're synthetic compounds. So there is a process by which B cells, individual B cells, can generate an antibody that is specific for basically anything. But because antibodies are proteins, they need to be coded for by genes, and having a gene in your genome for an antibody that can recognize any shape is just not tenable. So evolution...
How many genes are there?
How many genes are there total? I think there's there's between 20 and 30,000 in humans, I think.
20 and 30,000.
18,000.
Killer. That is not between 20 and 30,000.
You said 18? I've heard higher estimates than that. I mean, it's squishy because early estimates, I think right after the Human Genome Project came out, they were saying 30,000, and then a bunch of those turned out to be random bullshit, or they aren't actually thought to be coding sequences, but they were like open reading frames. Not all open reading frames are actually genes. So
sure, it's important to point out though that we, I think, haven't quite discovered all of them. So let's stick with the 20 to 30,000 number. And it was a bit of a leading question because the potential diversity just based in the genome, hard coded into your genome for antibodies, is somewhere in the range of 3 times 10 to the 11th.
Right.
So you have 3 times 10 to the 11th possibilities of antibodies, but we only have 20 to 30,000 genes, right? So you have to have a way to get all of those specificities into a single genetic entity.
Right.
So according to the National Human Genome Research Institute, we have approximately 21,000 genes. And I think the higher numbers go to the yeast and some of those other microbes. They start to get higher and higher numbers. At least some of them do.
So you're less complex than yeast.
Oh, yeah, definitely.
Congratulations. Well done, evolution. Okay, so basically, the way that this diversity is generated is actually kind of ingenious. So it turns out that the way this diversity is generated is through a number of mechanisms, but the simplest way to think about it is in terms of combinatorial diversity. So the part of an antibody that actually binds onto something, that actually grabs onto what we call its ligand, is generated by 3 different gene segments called V, D, and J for a variable, diversity, and junction. And the key is that there is more than one copy of V, D, and J. And I don't just mean like more than one copy, like we have 2 copies of every genes. I mean that on the chromosome that codes for the VDJ region, there are a large number of Vs, some slightly smaller number of Ds, and a number of Js. And those Vs, Ds, and Js can be rearranged in the genome. The DNA is actually cut up and re-spliced together to form different versions of this antibody, and this happens on a per cell basis. So every B cell picks a different V, a different D, and a different J to generate their antibody binding site. And it gets even more complicated than that. Antibodies are made up of 2 different protein chains coded for by 2 different genes, the heavy chain and the light chain. The heavy chain is based on recombination of these V, D, and J segments. The light chain has their own Vs and their own Js. They don't actually have Ds, but the same principle applies. You can sort of mix and match Vs and Js in the light chain, Vs, Ds, and Js in the heavy chain, and then one heavy chain and one light chain are going to come together and form your actual antibody. So we were talking before we started recording about potential metaphors for this to make it clear, and Matt suggested the idea of a 5-course meal, right, where you have an entree, an appetizer, a dessert, maybe a drink, all of these things, and you have a menu where you say, like, you can pick one of any 10 drinks, one of any 10 desserts, one of any 10 entrees, one of any 10 salads. And even though you have a fixed number of potential menu options, the different meals that you can generate is huge. And in actuality, it's much higher than that. I didn't actually look up the numbers ahead of time, but it's something like 20 different Vs. Is that right?
Yeah, it's somewhere in there. It's on the range of between 10 and 100. It's for each one of those. Actually, the J's might be a little bit lower than that. No, it's still probably within the 10 to 100 range. But
yeah,
I mean, the number that you come out with combinatorial is a great word.
It is a good word. But I don't think the combinatorial diversity alone gives you that 10 to the 11th number. There are even more. So you have this combinatorial diversity where you take V's, D's, and J's and you mix them together. But then you also have these additional mechanisms of generating diversity that we're not going to go into in great detail right now, but basically the way that these different segments are smushed together is slightly inaccurate. So you can get random nucleotides, random bits of the genetic code inserted into those junctions. So you actually end up with a huge array of different ways to put together an antibody gene. And like I said, every individual B cell does this separately and randomly. So in principle, every single B cell in your body is different, has a different antibody that's coded for at the genetic level.
And you have so many B cells too in your body, right? You have so many at any given time, and they're very short-lived. I think the rule of thumb that immunologists use, if you have a really complicated bug, right? Let's say flu. If you're looking at your naive B cell population and you're trying to ask what percentage of these B cells are going to recognize flu, the number that people use is something like 1 in 10,000. So for every complicated thing that comes in that attacks you, maybe only 1 in 10,000 B cells will actually have a receptor that even vaguely recognizes the thing coming in. This is necessarily a really low probability process, and it's an inaccurate process, but it does have the benefit of basically generating any specificity you could ever want. And if the B cell this time around doesn't see anything, it'll die and somebody else will take its place in 7 days. So it's pretty quick turnover too.
Right, and it's important to understand that when I get infected with flu and Matt gets infected with the exact same flu, maybe 1 in 10 or 1 in 20,000 of my B cells is going to recognize that flu. 1 in 10 or 1 in 20,000 of Matt's B cells are going to recognize the flu. But my B cell that recognizes the flu and Matt's B cell that recognizes the flu will have come up with a receptor that recognizes flu in a completely different way. I might have different alleles. We mentioned alleles last week. These are versions of the gene. So I might have different versions of V, D, and J. But even if Matt and I were identical twins, we had the exact same V's, D's, and J's, the B cell that I have that happens to recognize flu is probably going to be a different V, D, and J in the heavy chain, a different V and J in the light chain, and we're going to have a completely different response or a different antibody generated against that flu, but both of them are going to recognize the flu. And so that's maybe a good segue to talking about how these things actually function. So when you get exposure to flu, we're not going to talk about T cell help today, which is required. We're just going to talk about the B cells. But the simplest way to think about it is that you have a B cell that recognizes the flu. It gets the signal through its receptor, through its B cell receptor, which is the antibody basically attached to the outside. And it gets some other signals saying that there's an infection. And that B cell says, okay, it's my turn. And it starts to divide like crazy. And during division, it's copying its DNA, like all cells do when they divide. And because that B cell receptor is coded for at the DNA level, their actual DNA has been rearranged and recombined. Every time that B cell divides, it's going to make an exact copy of the same antibody receptor. It's going to have the exact same specificity as it divides. So you're going to get this
army of clones.
Right. We call this clonal expansion. So you're going to get thousands and thousands and thousands of copies of this same B cell receptor that we know recognizes the flu, because it wouldn't divide. It wouldn't get that activation signal unless it could recognize the flu. In addition, B cells go through an additional process to increase diversity. So what they do is they actually they actively turn on mechanisms to cause mutations at the site where antibodies bind to their targets. So you have a protein coded for by DNA that's been generated by this random specificity. So it's going to bind with some, we call affinity. It's going to grab onto that flu at some place on the flu virus with some level of specificity. As those B cells divide and make copies, they're going to introduce new mutations into that site of binding to the flu.
Into the VDJ region.
Right, into that same region of the antibody that's responsible for binding, that's diverse because of the recombination of these V's, D's, and J's. As those B cell receptor genes mutate, some of them, actually a lot of them, maybe most of them, are going to get less good at binding to flu. Some of them are going to end up with mutations that cause the protein to misfold. Some of them are going to be nonsense mutations, so they can't even make an antibody gene anymore. Most of those mutations are going to cause problems. Those B cells that lose the ability to bind to flu don't get to make new copies of themselves. But the few mutations, and there usually are going to be mutations that cause an increase in the ability to bind to flu, they're going to be able to bind flu better. They get positively selected. So there's sort of a mini natural selection going on with increased mutation at the site of binding and selection for only those mutations that increase the ability of the antibody to bind. So at the end of this clonal expansion, you've made tens of thousands of copies of this same B cell receptor with slight modifications, all of which increase the affinity. So at the end of this expansion phase, you have tons of antibodies that have really, really good, high affinity, good binding antibodies.
What does an antibody do once it binds?
What does an antibody do? So I've been talking this whole time about the receptor. So these are basically antibodies that are attached to the outside of B cells, and they can signal to the inside of B cells to say like, hey, we can bind really strongly. But then at the end of this reaction, you got lots of these high affinity B cell receptors. That B cell starts to secrete those antibodies. In other words, it starts to release tons of copies of these receptors from the surface of the B cell, and those antibodies can then go out into the serum, right? So or into tissues where infections might be happening. So remember, we talked about the fact that there's anti-sera in the serum of vaccinated animals, and I said that serum is blood without the cells. So there's no B cells left in serum, but all those antibodies that have been released from B cells are in the serum, and they can act to kill bugs in a couple of different ways. For viruses especially, or for toxins, they can bind to those and neutralize them, prevent them from being able to infect cells in the case of viruses, or from carrying out their functions in the case of toxins. Or they can sort of coat the outside of an infectious microbe and make it easier for other cells of the immune system to grab onto them, engulf them, and kill them off. In the case of bacteria, you can also have what I talked about earlier, complement. Again, we're not going to get into the details of this, but complement can actually directly make bacteria explode through a series of chemical reactions that punch holes in their membranes. So getting back to the original topic here, getting back to vaccination, the point of vaccination, or at least what most people think is the point of vaccination, is generating high quality antibodies to an infectious microbe. So going back to Jenner, when Jenner infected people with cowpox, what was actually happening is that those people were mounting an immune response that generated antibodies that could bind to the cowpox virus really well. And because smallpox was related to cowpox, those antibodies that could bind to cowpox could also bind to and neutralize smallpox. So when someone was exposed to the smallpox virus, those antibodies would bind to the smallpox virus and prevent infection, or prevent infection from spreading very far. So all of the vaccines that have been generated since Jenner are basically variations on that same theme. So we take a weakened or a killed version of a pathogen, we inject that into people, or we get them to inhale it, and those people generate an immune response that generates antibodies, and those antibodies are capable of binding to the virus or toxin and killing it or preventing it from infecting cells. And that's worked really well for a lot of diseases. I mean, the things that you get vaccinated against, we actually don't get vaccinated against smallpox anymore. And that's because the smallpox vaccine that Jenner and then future generations of scientists developed is so effective that we actually eradicated smallpox. There is no natural infection of smallpox happening anywhere in the world. So you don't actually have to get vaccinated anymore because the bug doesn't exist anymore. At least not in the wild.
Right. Except
apparently there's still some vials in CDC laboratories in Atlanta and some in Russia too, I think.
And also in the old Soviet Union, which is a little bit concerning. It's like, I think it's in 3 places. 2 are in the United States and one's in like Georgia or something like that, in some bunker somewhere in a BL4 lab.
Georgia, not the state.
That scares me.
Not Atlanta, Georgia, where the CDC is.
No,
Georgia,
not the
country,
state
former
of
Soviet
Georgia,
bloc.
the country of Georgia.
So anyway, we have basically we have had up until this point a sort of slapdash approach to making vaccines, and it's been super effective up until now. We have really effective vaccines for smallpox, really effective vaccines for polio, actually a couple different versions, one that uses a killed virus and one that uses a weakened virus. We have wonderful vaccines to a number of bacterial infections, things like diphtheria and tetanus, viruses that we mentioned, like the ones that cause measles and mumps. We now have chickenpox vaccines, although I didn't get one when I was a kid.
No, I just got the chickenpox. My mom put me in a chickenpox party.
Yeah, it's actually...
Thanks, Mom.
Speaking of anti-vaccine wackos, have you heard of the lollipop thing?
Yeah.
People getting their kid with chickenpox to lick a lollipop, and then they send it to another family, like through the mail sometimes, for their kids to... Like, how is that better than a vaccine? It just boggles the mind.
I don't know.
Viruses are natural, man.
Yeah, I agree with you. And like, so chickenpox has the benefit of
not killing
being unlikely to kill you, right?
Yeah, and it actually turns out if you get infected
our parents...
it made perfect sense for our parents to do it, because actually, if you don't get chickenpox when you're a kid and then you're exposed as an adult, it can actually really suck. It causes something called shingles that is awful. And I'm not actually sure why adults that get exposed to that virus, which is called, I think, variola, the variola virus, if you're exposed as an adult and you don't have any immunity, then you're kind of screwed. You got a really terrible disease. But if you're exposed as a kid, you do just fine. I don't know why that is.
Right, and shingles can even pop up if you've had chickenpox and then, as a kid, and then not had it for 40 years. Unfortunately, that virus has a tendency to sit in your peripheral nervous system, and it doesn't do anything for a very long time until one day it decides to do lots of things, and then it hurts a lot. So, all that being said, the chickenpox vaccine is effective now.
Yeah.
So, don't worry about getting it. It's good.
Yeah. Get vaccinated if you haven't already. Okay. So we have all these really great vaccines, and our slapdash approach to generating vaccines has worked pretty well for a lot of different infectious diseases, but we've run into a couple of different problems. And we're going to talk about 2 specific problems in vaccination that we've run into in the modern vaccine era, and some ways that scientists are trying to get around those problems. And so the first thing that we're going to talk about is influenza. And I think most people are familiar with the flu, but most people, when they get the flu, they have a really terrible disease and it sucks, but it doesn't kill you. But actually, influenza kills between 3 and 50,000 people a year in the US alone, every year, and more than that around the world. It causes major epidemics every year. Sometimes it causes what we call pandemics, so it's even higher spread, and it's really bad. It's a major public health concern. Influenza itself doesn't usually kill you. It's often secondary infections of bacteria in the lungs that kills people, and it usually kills the elderly and people with compromised immune systems, sometimes very young children. But it's actually a major public health threat. I mean, it sucks if you get it and you're young and healthy, but it actually causes a lot of death and disease around the world. So we have a vaccine for influenza, but as those of you who get your vaccines every year, and you should get them every year, know, you have to get them every year. And the reason for that is because influenza from year to year changes. The circulating strains of influenza, and there are usually a lot of them, change every year. And before flu season starts, a group of scientists gets together. They analyze data from different animal populations that can transmit flu to people. They look at strains of influenza that are circulating, and they try to figure out, they basically try to predict what strains are going to be most prevalent in the human populations in the coming year. And then they start vaccine development. They have to make vaccines. They have to start making the vaccines several months before they start administering the vaccines. So you can't wait until people start getting infected with flu to start making the vaccines. So this is a real problem. What we really need is we need a vaccine that can target many strains of flu at the same time effectively, so that you only have to get it once or only once every many years. And the other thing is that the way that we make flu vaccines, as I mentioned, takes months, and it's not 100% effective because, well, for a number of reasons. One being that the predictions of what the circulating strains of influenza are going to be are not always accurate. And because they're generated sort of in a hurry every year, we can't go through a lot of optimization. We can't make sure that you generate the best immune response against every strain every year. And so the actual effectiveness of the flu vaccine is typically between like 50 and 80%, which is good. It's better than nothing, but it's not nearly as high as we'd like it to be. So the first paper that we're going to talk about is looking at the sorts of antibodies that are really good at blocking influenza and how we can maybe get better at eliciting those types of antibodies in our flu vaccines. So Kate, you want to present this paper?
Yeah. So the paper that I chose is called Structural and Genetic Basis for Development of Broadly Neutralizing Influenza Antibodies. And the first author on this paper is Daniel Lingwood, and the last author on this paper is Gary Nabel. And I have to say that I love this paper. I saw Daniel Lingwood give a talk where he talked about this paper and some extension of this work about a year ago, and he was great. And the way that this paper is written, the story comes across very, very easily. And so I just really like this paper a lot. So generally, the vaccine for influenza, the way it works is that they induce antibodies to a certain region of the influenza virus. And so that region is the outer membrane protein called hemagglutinin, or HA. And normally, the way HA looks is it kind of looks like a lollipop. So there's a giant bulb at the top, and then there's a stalk that connects the protein into the membrane of the virus. And most of our antibodies are directed at the top of the lollipop. And the problem with this approach is that this area of the virus is quite variable. So when you hear about all the different types of influenza, like H1N1, H7N9, when they say H, they're talking about hemagglutinin, and then the number is the different types. So what we end up doing is making, like Kevin said, new vaccines every year. So we make vaccines against these different types of hemagglutinin, and those different types of hemagglutinin are variable. But what's interesting about the protein is that the stalk region is actually quite similar between all the hemagglutinins. And so the stalk is a part that connects the head of the protein to the virus membrane. And
It's the stick on the lollipop.
yeah, the stick on the lollipop. And this is called the stem region of the virus. And recently there's been like a lot of work into trying to make vaccines that target the stem. People think that this would be how we could make a universal flu vaccine. And so this paper starts with this observation that the stem antibodies, a lot of them come from one specific heavy chain allele. And so we were talking before about the different types of genes that are recombined to make the parts of the antibody. So one particular allele in a variable region of the heavy chain called IgHV169 comes up more frequently than if it were just by chance.
Right. And so that stands, that letter soup that you just sort of threw out, that's immunoglobulin, heavy chain, V region. What was the number there?
1-69.
Yeah, so you've got a certain number of V regions, and that is the 69th one.
So to use our previous metaphor, this is like every antibody that's good at recognizing influenza ordered the steak as part of their buffet. Every single one for their entree, they picked the steak. So they might have had a different dessert, they might have had a different drink, but all of them had the same entree. Or at least it seems like having the steak was way more common amongst antibodies that targeted the stalk than any other entree.
Right.
Just give us the metaphor, Kate. Just give it to us, okay? Just let us have it.
Okay. And so among the antibodies that have this heavy chain gene segment, these antibodies undergo really low amounts of somatic mutation. So I think they say in one of the antibodies, only 14 amino acids are changed compared to what was originally encoded in the DNA. So after the recombination, there's a process of somatic mutation where you guys can correct me if I'm wrong. Or maybe explain it. But during this process, there are some random mutagenesis that the antibody undergoes in the hope that the affinity of the antibody will increase. So these antibodies are really interesting because they don't need to undergo a whole lot of that mutation in order to bind flu. So they first identify if it's the heavy chain or the light chain of the antibody that is conferring the recognition of flu. So they make chimeric proteins where they have the somatic heavy chain or the germline light chain, or they have the full functioning antibody, which is a somatic heavy chain, somatic light chain, or they use a germline heavy chain and a germline light chain. And the thing that's interesting is that just having the somatic heavy chain is enough to bind flu. So the light chain is not making any considerable contacts with flu, or is not very important for the recognition and binding of the flu stem region. So what they then do is they make a couple more mutations where they basically take all the different places that one of the antibodies that they focus on differs from the germline. And so they make just all the different combinations to identify which changes are important. And they see that two regions are mostly important for the binding and neutralization of flu. And these two regions are called CDRH1 and SFR3. So the original antibody CR6261, it differs from germline in just 14 amino acids, but only restoring 7 gave the antibody its full potency compared to wild type to bind and neutralize influenza. And okay, so this is actually really cool because CDRH1 and FR3 are right next to each other in the folded protein. And they actually analyze different amino acid changes. And they find that in the folded protein, there's this phenylalanine, which is an amino acid that's quite large. And it's very hydrophobic, which means that it tries to avoid water. And phenylalanine 29 is flipped out. And it normally is buried inside the protein and like hidden from water. So phenylalanine doesn't want to be around water, so it normally buries itself around other things that don't like water. But in this protein, it flips out, and it's like very important for this to contact the stem region of the HA protein. There's another contact within this antibody that also helps confer like basically make a hydrophobic ledge. This allows the antibody to bind HA.
So what does this mean? So we know that there's this particular V region that's really good at binding flu if it just gets a few mutations. So why is that useful information to have?
So what's interesting is that there's no other antibody that has a CDR H1 that has a hydrophobic shelf. No other antibody does this. And so it implies that this was an evolved gene, that there's been like such great pressure from influenza that we have evolved a particular allele to recognize influenza. And the ability to bind influenza is encoded in our germline.
So that's really cool. So in a whole array of randomized antibodies, you're saying that one particular set of VDJs has actually been selectively evolved to specifically bind flu.
Well, one V, not even the V, D, and J together. It's just a V region that's
really cool.
Specifically evolved to bind flu.
Okay, so I actually said that wrong. It's actually the CDR H2, not the H1. So in the last figure, they look at the contribution of the CDR H2 region of the protein. So that's just one of the variable regions in this protein that's responsible for binding the antigen directly.
Right. CDRs are complementarity determining regions. So that's in the 3D folded structure of the protein, the part of the antibody that's coded for by the V, D, and J in the DNA. It's the 3D structure of the protein that's actually binding to the thing that the antibody binds to, in this case, flu.
Yeah. And so the second interesting thing, so not only is this like gene been somehow selected for, so we all have this, if you have this allele, you have the innate ability to recognize flu. So in the first figure, they talk about how the germline, when secreted, the secreted protein, doesn't bind HA. But in the last figure, they realize that if they change the context of how the antibody is being presented to the antigen, so to the viral protein, they can actually change the affinity of the antibody for the antigen. So in
the germline, you mean, so before any somatic hypermutation, if the context is right, this V region, if it's in the antibody, can actually bind flu innately before any somatic hypermutation.
Yeah, so the germline heavy chain and the germline light chain can bind HA, the virus protein, if it is an antibody that's bound to the membrane of a B cell. And so they show, so this type of antibody is called an IgM antibody, but basically it just means that it's in the membrane, and when the antibody is in the membrane, it then is able to bind this HA independent of all the hypermutation. And so this is really cool, that just like the context of the antibody changing allows you to bind HA.
So the antibody in the membrane of the B cell, that's the context that all B cells are going to first encounter influenza. Because like I said, B cells don't actually start the process of clonal expansion. They don't start the process of somatic hypermutation until they've recognized flu with their antibody bound to the surface of the B cell. So the B cell with the B cell receptor, the antibody in their membrane, is always the context of first encounter. So the fact that non-mutated antibody is capable of recognizing flu inherently is, it very strongly implies that this particular allele of the V region has been selected for recognizing influenza, which suggests that influenza has been a problem for people for a really, really long time.
I agree.
Those are the main conclusions of the paper. I think it's really interesting.
I think it's really cool because I guess we're reiterating at this point, but you have this system that's set up to be completely variable, right? Infinitely diverse. And so the whole basis of B-cell biology is essentially that you have this unbiased library system that is set up just so that you can potentially react to any molecule, any chemical ever. That's how our system works. And to find that there is one pathogen in particular, influenza in this case, that's been around in the human population for so long that we're starting to even bias that, right? So this is supposed to be an unbiased system, but we're biasing it slightly so that certain parts of the B cell repertoire inherently recognize flu better at baseline. I actually think that that's a really cool finding.
Yeah, but I don't think it's inconceivable to think that there might be other examples of this. I mean, even in a system that's designed, we should say designed in quotes, evolution has selected for a system that allows infinite variability. It's not terribly surprising that within that ability to have lots of variability, there would also be some bias towards pathogens that historically we've been exposed to a lot. So it wouldn't surprise me if people found other examples of this sort of thing for other pathogens that have been around for a long time.
Yeah, and
Totally.
and maybe they're important for the pathogens that like we don't. So the hope is that maybe there's some sort of germline that would be preferential for HIV or SARS or like these viruses that have like great public health importance. But I mean, it's unlikely to have already been selected because our interaction with those viruses like hasn't been long enough. But perhaps this is like a general mechanism or something that's more common when we have the viruses that have been around for a long time that we've evolved towards, that they've evolved towards us, and that it's one of like the checks and balances. You know, like something like herpes or something like that, like something that is very common and, you know, doesn't give us a whole lot of disease anymore.
So does this in any way help us to make a better flu vaccine? This knowledge, the understanding that this particular V region of a B cell or of a B cell receptor, of an antibody, is really good at binding to flu, does that help us to design a better flu vaccine that would mean that maybe we don't have to get a flu vaccine every year, or a vaccine that's more effective?
Yeah, so these antibodies are interesting because they're against a different target than the normal flu vaccine is. So it's against the stem of the HA protein instead of the head of the HA protein. So like the stick of the lollipop instead of the top of the lollipop. And so we don't have any vaccine that targets this part of the protein. And the concept that it's more conserved is appealing because that means that the vaccine would be effective against many types of flu instead of just one type, and it would eliminate the need to make a flu vaccine every year.
Is there a reason that they hadn't tried? I mean, they must have seen prior to this that region in the flu genome was more conserved. Is there a reason that no one has really tried to target that region before?
I think a lot of people have. It's just really hard to...
I think it has to do with the way that we vaccinate for flu. So the way that we vaccinate for flu is we grow up, you know, like these attenuated strains and kill them. And then we, you know, inject killed virus into a person's body. And I think that it might just be a bias of the immune system to target the head as opposed to the stem.
So maybe it's the easy target rather than the most effective target. So your immune system takes the easiest route.
But yeah, let's switch over to the HIV so I can finish my lab work tonight at some point.
Okay, go.
So the next paper we're going to talk about is not influenza anymore. We're going to be talking about HIV, human immunodeficiency virus. And I'm sure that most of our audience has heard of HIV before. It's the causative agent of the AIDS epidemic that was first recognized in 1981. The first AIDS patient was recognized in 1981. And then the virus that causes AIDS, HIV, was discovered in 1983. So 2 years later, scientists are really good at generally at determining causative infectious agents of diseases. And when the virus was discovered, scientists were pretty thrilled and thought, all right, great, we're going to make a vaccine. Just give us a couple years. We're good at this. We know how to make vaccines against viruses. And so here we are 30 years later, and we still haven't made an effective HIV vaccine. And that is certainly not for lack of trying. Lots of money is being spent trying to make an HIV vaccine. Lots of labs have tried, lots of different clinical trials have gone through, and we recently had some success with an HIV vaccine. I think it provided, what was it, Kate? It was something like 30 or 40% protection for a couple of years.
I refuse to say that there was any success in the vaccine trial RV144.
Okay.
If the statistics are really, really...
Oh, that's right. I forgot that you hate these statistics.
No, I don't hate the statistics. The statistics are weak. I'm just saying, like, if there was one miscount, the significance of that vaccine study would have been nothing.
Okay, so there is one vaccine that people have claimed is a success, but it's tenuous at best. And do we want to cut that out, Kate? Should I start this intro over so that you don't have to be on record saying that you think that...
I am fine being on record saying that RV144 is not a successful vaccine. It's not. If it were a successful vaccine, we would be using it right now. But we're not.
All right. Well, there you have it, folks.
I like it. That's conviction right there.
So we suck at making an HIV vaccine, even though we've tried a lot. So the next paper we're going to talk about is basically trying to use the fact that we know that antibodies for many viral infections are good at providing protection. We also know that there are some people that make what we call broadly neutralizing antibodies against HIV. So one of the problems with making an HIV vaccine is that it's even more variable than influenza. Within an individual person, even, you can have tons of different strains of HIV. So even if you make a really, really wonderful antibody against the HIV that is infecting you, there are probably lots of other copies of HIV in your own body that are going to evade that antibody. But there are these broadly neutralizing antibodies that some people make that do seem to provide at least some protection. It's not necessarily that these people are immune, but they at least have some protection. So the second paper is basically using that knowledge of the presence of broadly neutralizing antibodies, the fact that antibodies can provide some protection, and the fact that vaccines, because people generate antibodies randomly, it's really hard to get people to make specifically the antibody that you want. Because even though one person has these broadly neutralizing antibodies, a specific V, a specific D, a specific J, with all of the hypermutations, it's basically impossible for us to plan that. So this paper is taking a different approach. It's not a vaccine exactly, but it's using those principles that we've learned from vaccination to try to protect us from HIV. So Matt, why don't you take it away?
Yeah, so one of the really hard things about HIV is that it is a really random virus that's just made its way into the population. So HIV has not been around in humans for a long time. Going into that, the name of the paper, or the title of the paper, is HIV Therapy by a combination of broadly neutralizing antibodies in humanized mice. So I'll talk about all the individual components there, but the paper is out of Michel Nussenzweig's lab at Rockefeller, and Michel is actually not an HIV guy. He's sort of recent on the field, and I was at the conference that he unveiled this paper, and I was just really, really impressed by it. He's a fantastic scientist. He has a great lab, but he's not a traditional HIV guy. He's actually a VDJ recombination guy, and he is from a proud lineage as the Cavendishes, I suppose, and maybe we introed on this. The Nussenzweigs are also a family dynasty, so both his parents were pretty big scientists. Victor Nussenzweig, the father, is a pretty famous scientist as well. So they've been doing this for a little while. So where this paper sort of starts is you have a B-cell biologist who's really well versed in VDJ recombination. He's necessarily really well versed in antibody production. Is taking a look at the field, the HIV field, and saying, well, it's sort of unfortunate that antibody therapies haven't worked in the past, because previous people have suggested that if you take a patient that has HIV and you give him antibodies against HIV, the HIV will potentially respond. Maybe the virus levels will go down, but eventually the HIV is going to come back. So the HIV is going to find a way around the antibody, and it's going to respond back. So Michel's group, the Nussenzweig group at Rockefeller, decided to take a different approach. As Kevin mentioned just a couple seconds ago, most people get infected with HIV, and unfortunately their immune system doesn't really respond all that well. But for a select few people, over the course of many, many years of infection, I mean, you're talking in some cases decades of infection, their immune system starts to kind of figure it out. They still acquire the disease, but they have these antibodies that are considered broadly neutralizing antibodies. In this case, what we're talking about is patients who have been infected with the virus for years and years and years. And over those years, the B cells are continuously dividing and continuously dividing and continuously mutating. And in some very select cases, the antibody gets really, really good at recognizing HIV. And so similar, actually, to the way broadly neutralizing antibodies to influenza can arise, those same sorts of antibodies can arise in HIV. It just happens at really, really low frequency. Now, the key is, in the case of HIV, even if a patient after 15 years develops one of these antibodies, the virus is still good enough to mutate around that. But what Michelle Nussensweig decided to do is go out into the population, collect all of these different broadly neutralizing antibodies, and just start to play around with them. And when I say play around with them, I mean, as an immunologist who works on antibodies, understand where the antibody is actually binding on the virus, what the affinity is, and most importantly, he took a look at these antibodies, he looked at where they bound to the HIV virus, and he went in and he engineered and he made the antibodies better. So whereas you have these broadly neutralizing antibodies that arose after 20 years of constant mutation and now are sort of good at broadly binding HIV, he basically went in and made those antibodies, in some cases, 100 times higher affinity towards the HIV virus. So you're talking about the smart engineering of these really potent antibodies that exist in the human populations at really low levels. So essentially he went out and he found 5 of those. And so how did they actually test these antibodies? It's really hard to test antibodies against HIV because the models are really difficult. So unfortunately, whereas I can do all of my experiments in mice with influenza and the vaccination process, the reason that I can do that is because mice actually get the flu. Mice get influenza, they get sick from it, they die from it. It's actually very similar to the way that humans get the flu. Unfortunately, mice do not get HIV. And the reason that they don't get HIV is because they don't have the appropriate receptors and...
So HIV can infect mouse cells. It can actually even integrate. It's just that the proteins that are needed to express HIV, they're highly evolved and they're highly specific for the human transcriptional machinery. So it actually just can't use mouse transcription machinery to replicate. So you can use HIV, that's why you can use HIV-derived vectors in research and infect mouse cells, but there's no expression off the promoter of HIV.
Okay, so you get infection then, but unfortunately you don't have a mouse that will develop AIDS. So you can maybe study the virus itself and the process by which it integrates into a cell, but maybe not the downstream disease.
Yep. Yeah, so there's no productive replication of virus.
Cool. And yeah, this is why it's good to have an HIV person or someone that at least knows what they're talking about. So it's really difficult to study HIV in a mouse system. You really have to go with either a human or a non-human primate. Or, and this is what the Nussensweig group decided to do in this study, you can go with a combination of 2 things. So instead of approaching it from an angle where you're trying to actually infect humans, because you can't infect humans, that's not ethical even a little.
We could, in principle, infect humans, but no IRB panel would allow.
Yeah, it's not going to. The protocols are already hard enough to get across the board when it's just mice. Human infection trials are really difficult to convince people to do. What you can do, though, is you can take a mouse that is primarily deficient. It has no immune system. You basically take a mouse that does not have any B cells. It does not have any T cells. Basically, these mice have no functional adaptive immune system. So that's the background that we're talking about. What you can do with those mice is, at birth, even though they have no functional adaptive immune system, you can take human fetal liver tissue. And human fetal liver is actually the precursor immune organ to what will eventually become the bone marrow. So initially in humans, the entire hematopoietic system starts in the liver. It will migrate to the bone marrow, and so that's where all of your hematopoietic cells will start. So
let's just say hematopoietic stem cells are the cells that give rise to all of the cells in your blood. That includes T cells and B cells that we've been talking about today. It includes macrophages, red blood cells. Basically, all of your immune cells and blood cells are formed from the hematopoietic stem cell compartment.
Right, which in humans, in adults, it's all in your bone marrow, which is why you give people bone marrow transplants, right? If you're trying to reconstitute their immune systems, you give them bits of your bone marrow.
We talked about this a bit on the last episode, too.
Yeah, exactly. Go back and check out episode 1. But if you're talking about fetal humans, so this is early on in development, that entire hematopoietic system is still in the liver. So if you take pieces of that liver and you transplant them into mice that do not have an adaptive immune system, what you get is you get a mouse that has what is essentially a human immune system. So the human immune system sets up shop. The mouse doesn't know any different, and so as the mouse grows up, but it has a human immune system. And the benefit of that in the HIV field is, of course, that if you infect that mouse now with HIV, the HIV can take advantage of the transcription machinery that it needs in order to produce a productive AIDS course. So instead of getting, this non-productive infection, but no new virus, you basically get the reconstitution of the full disease in mice.
And it should be pointed out, as in all cases, so this is a model of HIV infection, and all models are fraught with peril. This one is particularly fraught because it's a human immune system in a mouse, which is all kinds of crazy. But it's much better than a lot of other models that we have, or it is better in some ways than other models.
Yeah. And so the other major model that's usually used outside of straight human trials is non-human primates. But there's actually a really specific, and they point out in the paper, there's a specific reason why they can't use non-human primates in this study. And the reason is that a non-human primate is just that. It is non-human.
So actually, just before we get too far down this road, a year after this was published, they published this exact same study in macaques.
Yeah. So you're right.
But is it the same antibodies? Because it seems like, so they respond to SIV as well?
So they use SHIV, which is an SIV-HIV hybrid. So SIV is the HIV equivalent for macaques, another non-human primate. Yeah, so they use these. The SHIV has the envelope from HIV, but has a lot of the parts of SIV, so it's able to replicate in the monkey. And they use similar antibodies to this one. So some of them they use the same. Some of the antibodies that they use are the same ones in this paper.
Okay. So the antibody has actually 2 regions, and you can take a look at the diagram on Audiommunity, but basically you have the region that recognizes whatever it is the antibody's against. So the part of the antibody that recognizes HIV, and that's the variable region, because that's different between all antibodies. But you also have this other region called the constant region, and the constant region is just that. It's constant between all antibodies. So usually when you take an antibody, for example, against something in a mouse, so if a mouse makes an antibody, it has a constant region that has a mouse signature on it. So if you want to use that in humans, you have to turn that constant region into a human constant region in order to use it. And I assume that the same would be true if you want to use these antibodies in macaques. And so I'm wondering if they had to change those constant regions into macaques.
So I looked into the article, and I'm not sure that they did, because they talk about re-injecting the antibodies in the macaques, and they mention that there has been an immune response against these antibodies, but that the antibodies are still effective, but just to a lower, the potency. The potency isn't there.
So maybe if this turned into a therapy, they would just go ahead and change that completely over to a humanized model. That's cool, though. That's cool that there's some species cross-effectiveness there.
Well, it's not species cross-effective. Well, I guess maybe because the types of antibodies are different, but the antibodies are for the same HIV protein. So, it's
what
still
I mean
HIV
is that
protein.
The same constant region.
Mm-hmm.
Yeah, the constant region is what I'm talking about, I guess.
Mm-hmm.
Well, so even that monkey model, because you're using a modified HIV, you're using the SHIV virus, that's not a perfect model either. So all this complicated crap that we're doing with this humanized mouse model means that this is not necessarily a perfect system to study it, but it's among the best we have. There are a couple of models for studying HIV. Some people use a different virus called simian immunodeficiency virus that's related to HIV in monkeys, and that has provided a number of good studies. But the only way to study actual HIV without infecting people is to put it in these humanized mice.
Yeah, but I think the point is, you use these mouse models as best you can, and in this case, you've got a mouse with a human immune system, and you use non-human primates when you can, and hopefully just when the science warrants it. But it's not perfect. I will go out and say though that I really do like the employment of this model in this study. I was happy with the way that they did it, so I wouldn't want to cast aspersions on this on the outset just by saying that the model is poor.
And I think this model's improved a lot in the last 5-10 years, because I remember you couldn't keep them out this long with HIV, but now it's 120 days, 4 months. That's great.
Yeah. So we've got this system which isn't perfect, but it's become better established over time, and they do a pretty good job in this study, I think, verifying that at least you can get what's called an acute infection, and that's as the virus is sort of wreaking havoc in your bloodstream. It's infecting everything that it can find, and over the course of that, it will be integrating into your CD4 cell's genome. We've got an animation that will go up sort of showing you exactly how that happens, hopefully. So they do track, and they do a pretty good job of showing that the model that they're using does give you a normal acute infection in the mice, so at least the infection sort of looks like it would in a normal human. And then if you track the amount of virus in the blood over time, it maintains pretty persistent levels. They track CD4 to CD8 T cell ratios, which is something that's really important in the HIV field as far as how far the infection has progressed. And one of the most important things, I think, for this study is they actually take a look at the genetic diversification of the virus in these models. And the reason that that's important is every patient that gets HIV basically has a mini evolutionary cycle that goes on with the HIV virus. So there's a constant mutation battle between the immune system and the HIV virus over time as you're infected with it. Basically in such a way where your B cells are trying to keep up. So your B cells are constantly trying to make an antibody that will attach to the virus, and the virus is constantly making new forms of itself that the B cell receptors and the antibodies don't recognize. So what they do in this study is they basically show that in these infections, in humanized mice, that's what they're called, is humanized mice. In these humanized mice infections, the virus is mutating like it normally does in a human patient. So that sort of brings us to, I guess, what the theory of this study was. And we talked a little bit about metaphors, and imagine you are a kingdom, right? You're looking over your land, and all of a sudden a new kingdom pops up. And that kingdom has lots and lots of different castles. So all of a sudden you look around and you're just surrounded by different castles, right? And you need to kill them all. You need to knock them all down and reestablish your territory. But not all those castles look identical. Some of them have moats, some of them have parapets, for those of you that are up to date in your castle warfare. So there's lots of different features of these castles. They don't all look the same. So you decide you're going to build your army. You need to build your army. You need to go out and you need to attack this kingdom that's filled with castles. And so you get all of your infantry together and you decide to march on all of those castles. And it works for the most part. The problem is that 10% of those castles had moats, and the 10% of castles that had moats didn't die. Your infantry was prevented from getting to the castle walls. And the next day when you wake up, the builders that built those moats, that built the castles with the moats, recognized that that was pretty effective in keeping away infantry. So now the same number of castles exists because they built all new castles, but now all of them have moats. And this is sort of what's happening when your antibodies are trying desperately to recognize HIV. You have an antibody that recognizes a specific part of HIV, and 90% of the HIV in your body has the thing that that antibody recognizes. And so what you get is a killing of 90% of the HIV virus, but the 10% that didn't get killed by that antibody is looking around and saying, well, that was a pretty effective thing that I did there. And so, it's going to be selected for in a way that the next morning, in this case, the viral replication cycle is actually much faster than that. All of the HIV in your body is going to have incorporated that mutation that made you completely resistant to the antibody. But imagine now, instead of just having infantry to attack all of those castles, you have multiple weapons. You have cavalry, you have trebuchets, you have all of these different things that people use to attack castles, and you use them all at the same time. So when you attack, some of them have moats, but even the ones with moats, even if they don't get killed off by the infantry, they get killed off by something else. And so at the end of the day, there are no castles standing. And that's sort of the idea behind this HIV vaccination therapy, where you have different antibodies. They all sort of represent, in this metaphor, a different weapon against the HIV. In this case, we have 5 antibodies total, but each one of those weapons has a very different target. It attacks HIV in a different way. And the idea is, if you use them all at the same time, the HIV is going to be killed abundantly right up front, and it's not going to have the time to recognize each individual antibody and find a mutation around it.
And I want to say that this principle is also in use in current HIV therapies. So currently, when people are infected with HIV, we give them a cocktail of drugs that help prevent HIV replication. And we don't just give them one drug, because if you just gave one drug, this exact same process would play out. It's not antibodies in this case, it's antiretroviral drugs. But the same idea applies in that if you attack HIV on multiple fronts, it makes it much harder for those viruses to evolve. Escape mutants is what we call them.
Right, and so they show this. Basically, they set a baseline at the front end of this paper, and they go through kind of what other people have done in the field, which is an idea called monotherapy. We have this broadly neutralizing antibody that we isolated from this patient. We made it better than it was, and we injected it as a single kind of antibody into the patient. And similar to other studies, what they found was, although a couple of those antibodies seemed effective, they were only effective for a very short amount of time. So within 14 to 16 days in every single mouse that they used this treatment on, the virus had almost completely rebounded and they were overrun again by HIV. If you sequence those individual virions, what you found is if you treated with antibody A, the virus used mutation A in order to get around it. So there were very specific mutations that the virus would make. It's almost like chess, right? The antibody makes one move, the virus makes exactly one move to counter. And so this process played out with every single antibody that they tried, and this was the problem with previous therapies trying to use antibodies. And it's true that some antibodies allowed the virus to make more general mutations. Maybe there were 4 or 5 chess moves that would allow the HIV to win, and some antibodies really required that a single mutation would arise that would allow you to sort of beat the antibody binding. So then they went into, I think, what is the more important part of this paper, which is the tritherapy and the pentamect. So tritherapy means that they took 3 of those 5 antibodies that they had isolated, and they started injecting those into mice, and they started to say, how did HIV do with 3 antibodies now? And in this case, we saw viremia decrease in 11 of 12 mice. So all of a sudden you go from an antibody therapy that is completely ineffective in the case of monotherapy, which is what had been done before, and now 11 out of 12 mice are responding. And it's true that only 3 of 12 had prolonged and effective HIV control, but it's still 3 of 12, right? And when we talk about prolonged and effective HIV control, what I mean is if you release the treatment, so you stop giving those antibodies, it's only after the antibody goes away that the virus comes back, which means that the antibody was essentially keeping the virus level at bay, which is a really, really cool finding. It essentially means that in 3 of those 12 animals, the virus had been essentially stopped in its tracks, and it had entered what we call the latent period, which means that it's sitting around in your genomic DNA, but it's not capable of producing virus that's going to spread to other cells of your body.
But it's also important to note that these mice aren't cured. They haven't eliminated the virus from their system.
No, but they have eliminated virus from the infective phase where cells in your body are transmitting the virus to other cells of your body. Essentially, what you've done is you've taken the virus from an acute infection and you've turned it into a chronic condition, which is much, much more controllable.
I'm not sure that they show directly, though, that cell-to-cell transmission is not occurring.
So when they allowed virus to come back, following in the 3 mice that showed long-term viral control, they then allowed the virus to come back, and then they did sequencing on the individual virions, and all of those viruses that came back only had mutations that would allow them to get around either 0 or 1 of the antibodies that they had shown.
Yeah, so what I'm thinking about is virus that is not going through a cell-free stage. So there's cell-to-cell transmission of HIV. So I think that the current theory is that there's some sort of like synapse that's occurring between 2 cells, so they're very, very close together. So the HIV buds out of one cell and then immediately infects the neighboring cell. So there wouldn't be enough time or space for an antibody to actually get there.
Right, and that could certainly be the case. But one thing that they do point out, and I think it's important, is that the HIV particles that do come back in those mice, they were still sensitive to the antibody treatment. Mm-hmm. Mm-hmm. So you're still not getting, the overwhelming infection that we usually experience with HIV/AIDS, which is really cool. So you can imagine if 2's good, 5's probably better. And this is where the paper really picks up steam, and this is where it gets really cool for me. So we talked about the reason that you use multiple weapons at the same time. If you use 5 and you run out the math, the chances, theoretically, of getting an HIV that mutates all at once around all 5 is just astronomically low. So you would expect that this would be an incredibly effective way of approaching this sort of problem. And so essentially, they just give it everything they've got. They inject these mice with all 5 antibodies after they've been given HIV. They injected 14 mice. Within 6 to 7 days, all 14 mice showed decrease in viral load down to an undetectable level. The 11 that survived the treatments showed huge control to below detectable viral levels for up to 60 days following administration of the pentamix, so the 5 antibody mix. And so they allowed these mice to get treated, virus drops down to baseline, and then they take away the antibody and they track how much antibody is in the blood. The virus only comes back when the antibody titers go down to a really, really low level. In some cases, that's up to 60 days. And if you take a look at the virus that comes back after those 60 days, after the virus or after the antibody levels have dropped to 0, the virus that comes back is just as susceptible to the 5 antibody mix as the virus that started. So essentially what you're showing is that this antibody mix is effective at dropping HIV down to undetectable levels, and it completely prevents the virus from mutating around the treatment. And that's where I think it's really, really cool. And it's actually an important little tidbit, a fun tidbit, actually, where during this entire treatment process, when HIV was being repressed, essentially, by this antibody mix, they were trying desperately to find HIV in the blood. And they did, a couple times, find HIV in the blood of these mice. But what's interesting about that is the HIV that they found did not have the protein that the antibodies targeted. So essentially, in order to try to outmaneuver the antibodies, HIV was producing virions that completely lacked the machinery in order to infect new cells. So,
Were they stop codons in the envelope?
Yeah, they were stop codons to GP120.
I bet that those type of virions are produced in normal HIV infections. So HIV has boatloads of mutations when it replicates. So there's probably these defective virions being produced under normal infections. It's just that they can't go and infect other cells, so you don't find a lot of copies of them.
Mm-hmm.
Right. That's almost certainly true in this. And basically what they're showing is the only viral particles that are not killed off by this treatment are the ones that are completely defective to begin with.
Yeah.
So this is why I put money on this antibody therapy. To be honest with you, I like this study, but more importantly, I like the evolution aspect of this study.
So why would you want these antibodies over antiretroviral therapy?
So they're actually doing studies now as far as antibodies in conjunction with antiretroviral therapy. And I think for the same reason that this study showed that taking multiple approaches to the same virus at the same time is more effective than just one, I think that this could be a really good way to supplement antiviral therapy.
I just want to point out that in order to get this therapy in use in humans, basically it would require infusions of antibody. So there are antibody therapies that are currently being used for other diseases. They all require injections. You can't just take a pill and have these antibodies in your bloodstream. You actually have to go and basically sit with an IV drip periodically to get this. So it's not the easiest therapy. It's probably not super tenable for places where HIV is a huge problem, like Africa and Asia.
So I actually disagree with that. And here's the reason why I disagree with that. You asked why I would want this in comparison to heart therapy now. Mm-hmm. And the reason that I would say is that one of the major problems with heart therapy is that the drugs are complicated and they have to be taken at extremely specific times.
Well, Truvada, I mean, I feel like that is starting to not be a big deal. Like
you're right.
Truvada is like you have combination pills now, but I agree that the timing can be tricky.
The timing is tricky, and more importantly, in my mind, if you get an HIV antibody infusion, what they're basically showing in this study is that there are some cases, and you could imagine that you could expand this repertoire of antibodies and basically give a polyclonal injection. Mm-hmm. I see no reason why in the future you couldn't have a monthly injection of this sort of thing. So rather than having an entire population that has a hard time with time and pills in general, I think an infusion is a potentially viable way of approaching this.
But you know what's actually something to note here?
Well, it is now.
Is that the sorts of viral suppression you see with this therapy is nothing compared to heart. Heart puts you to undetectable levels. Heart suppresses you 5 logs. I mean, they showed some people get levels. But you look at the suppression; it's like 3 log suppression, and it's so variable. I don't know if I would be giving up on heart just yet.
I'm not.
Well, I mean, if heart is already going to be suppressing you to an undetectable level, and the variability is so much tighter,
right? But imagine if you, on top of heart, so let's say that you use heart to knock down viremia to undetectable levels. Right? Mm-hmm. And then you have 3 broadly neutralizing antibodies that sits on top of heart. And if you forget to take those pills and the viremia comes back within 30 days, those antibodies are basically sitting there preventing that. So not only are you preventing mutation against the antibodies, but you're also preventing the mutation against heart. So I don't necessarily see it replacing anything, but I do see it as a viable way to supplement where the half-life of an antibody is much longer than those drugs. And the other thing that I would point out is that, yes, we're talking about antibody infusions here, but there are other labs that are working on ways to make these sorts of things expressed on a daily basis in a human subject, right? So David Baltimore's lab right now is super focused on this idea that you can take essentially genetic material, inject it into a person, and that will allow that person to create these antibodies from scratch. So now imagine in 20 years when we have a way to stably do that. What if you could take some of these antibodies, these mixes, you could inject the code for them into a person, and so that person all of a sudden is making 5 different antibodies that all fight HIV at a reasonable level. And is it as effective as heart? No, it's not. But it's still effective enough to control HIV outbreaks. So that's really the potential that I see.
So I think one of the reasons why people are so excited about the antibody therapy, even though it's not that impressive compared to heart, is the idea that the antibodies might target infected cells and kill infected cells, that there might be more functions to these antibodies than simply neutralizing virus. And so a lot of people keep invoking this idea, that there are latent reservoirs, or there are cells that have a long infection with HIV. So cells that will keep producing HIV and not die for weeks to months, and that maybe antibodies could target those infected cells and lead to the death of the long-lived reservoirs of HIV, which would be really interesting if that were true. I think they keep invoking this idea, but they haven't. But over the last couple of years, they haven't looked at it specifically. Like that would have been I think that would have been a more interesting follow-up than the monkey studies. But maybe you just need the monkey studies to get to your next phase.
Yeah, well, I think you show something like this and people immediately want it to be shown in humans, right? And the only obvious way to do that is immediately to go into non-human primates, whether you're ready for it or not. But sort of the reason that I'm really drawn to this paper, what I find fascinating about this is a reoccurring theme in human health. And so one of the problems that I have been really interested in the past is the idea of antibiotic resistance. So antibiotic resistance, and we won't talk too much about it here, maybe we can talk about it some other time, occurs in exactly the same way that HIV mutation occurs, in that you treat with one antibiotic and the bacteria finds a way around it. And so sometimes we use antibiotic cocktails, is what we call them, which is multiple different approaches at the same bug, but we don't usually do it. And I don't know why we don't usually do it. So I just feel like this is a step in the right direction about how you think about how the immune system and the virus, in this case, sometimes bacteria, are interacting on a day-to-day basis, and recognizing that if you really think hard about the evolution of that interaction, you can sort of jump ahead of it, predict what's going to happen, predict the sort of mutations that are going to occur in the virus, and maybe create a therapy that uses those predictions and keeps everything in check just a little bit better and a little bit longer.
All right, so on that note, I think we're going to wrap it up here. This has been Audiomunity episode 2 from emunity.org. I'm Kevin Bonham with Matt Woodruff and Kate Franz. You can find us on SoundCloud at soundcloud.com/audiomunity, and I'm just putting together our RSS feed, so really soon we're going to be on iTunes, so you can look for us there. You can find me on Google Plus and Twitter. We're on facebook.com/audiomunity, and you can find all of those links and links to our email address if you want to give us some feedback. We would love your feedback. You can also leave comments at emunity.org. That's EMMUNITY.org. We're supported in part by Harvard Medical School, and the music at the beginning and the end of the podcast was composed by Rachel Rennick. That's Audiomunity, and we'll see you next time. Thanks for listening.